Good afternoon, everybody. My name is Ove Nilsson, and I will be your host for this session of Worth Knowing today. First of all, I would like to welcome you all to this lecture. This is the second of 4 lectures this fall in the series Worth Knowing. The next lecture will be on the 14th of November when Mats Nilsson will speak on the topic The Land-Atmosphere Exchange of Greenhouse Gases of Boreal Ecosystems Under Climate Change. And the last lecture will be on 28th of November when Frauke Ecke will speak on the topic Varför biologisk mångfald är bra för vår hälsa, in Swedish, I guess. So before we go on to the main event, I would like to inform everyone that this lecture will be filmed and broadcasted so that you are all aware of that. The lecture will be about 20 minutes, and after the talk there will be time for questions. And for that we will use this microphone that I'm speaking in, so please make sure that you ask your question into the microphone so that we can document and broadcast also your questions. And now over for this day's lecture then. And it's my pleasure to introduce today's speaker, who is Professor Harry Wu. Harry Wu is a forest geneticist. He has a long career working in Canada, working in Australia with forest genetic questions and very often a very close connection to operational tree breeding also. Since 2010, Harry is also a professor in forest genetics at the Department of Forest Genetics and Plant Physiology at the Umeå Plant Science Center here in Umeå. And Harry is actually Sweden's only professor in forest genetics. So he will speak today about some exciting new technologies that has the potential to really revolutionize the way we are doing operational tree breeding. And the title of his talk is Genomic Selection, the Future of Tree Breeding? ? Please, Harry.
Thank you very much for nice introduction, Uwe. So good afternoon. Welcome to this my sort of veta so the sessions. And today I will talk about this new tools in tree breeding and the genomic selections. So I will first introduce you some of the general and the process of tree breeding. Some of you, if you are not familiar, so I will go through some of this basic process of tree breedings. Tree breeding is a cyclic process. So we usually start off selection of elite trees. We call sometimes plus trees from natural stands or from plantation. We usually select many trees from 50, maybe 300 for one breeding population, for one areas. Then we organize these breeding populations. Then we make a cross among those selections. So then from the seeds, we put it into the field for progeny testings. Then after many years, particularly for conifers, then we make evaluation, make a selection again. So those best one, we make a second generation breeding populations, and the very last trees, we're going to for propagation, for improved varieties or families, individuals, for new plantation. So this is the general process of tree breedings. Yeah. So this is the Norway spruce breedings in one breeding zone here in Sweden. There's many breeding zones. In one breeding zones, usually about they select the 50 best individuals from that breeding zone or nearby breeding zones. Then cross among these 50 trees. Usually one tree is crossed with another 2 parents. So that will create about 54 superfamilies. The cross take many years. So here I only put 4 and 6 years. Sometimes take more than 4-6 years, depend on the flower conditions. So from this 54 superfamilies, then within each families, about 40 trees will be propagated. So we call 40 clones So total will be about 2,000 clones will be propagated, and with each about 14 ramets. That means 14 copies from individual trees. So total about 28,000 trees cuttings will be tested in the 4 test sites within a single breeding zone. Sometimes maybe more than 4 test sites. So after like 12, 15 years, then we make selections, 50 best trees, each one from individual families, and make another breeding population. So we come like same circles. And now there are some motivations, but this is basically a principle. And the Skogeforsky, they do breeding for Norway spruce. So one question is, it's very long times for selection. We need to wait for many, many years for progenies. I think I should come to this ways. So the problem is, after progeny testing, we need to wait many years. So this is one of biggest constraints in tree breeding, is long-term progeny testing, particularly for conifers. So, however, we have to do some conifers completed the 4th cycle breeding a few years ago when I left CSR Australia 80 years ago. So they went to the 4th generation breedings. They started from 1950s. So, but radiata pine and growing much faster than our Norway spruce. So within like 60 years, we had almost 4 generations. So average about 15 years, 1 generation. But that's still too long. Because if that's continue, then one career of tree breeder like me, I can only have 2 to 3 generations. Compared to the crop, like rice, maize, you can have 1, 2 generations each year. So our breeding cycle is too long. That's the biggest constraint. So this is typicals and conifer breedings. And we need usually make several years crossing, gather the seeds. Then depend on species, we need progeny testing from 6 years to 15 years. Like radiata pine with 6 years, some of here northern species we need 12, 15 years. Then we need to evaluate selections. So total we need like 1 or 2 decades for 1 generations. So it's a really long times. I think some people here already, I know several tree breeders, they were doing this very good the last 20, 30 years. Only one made 1 or 2 generations. But that's already good contribution, but we are not satisfied with such long-term cycles. So how long then should we wait for selections? We are usually, and progeny testing, we can like from seedlings earlier to maybe selection age, but that's too long. Like for radiata pine, rotation age is at the 30s. We cannot wait for 30 years. For rotation age like Norway spruce, in southern Sweden's, like maybe 60 years. Here, maybe longer. So we cannot wait. Otherwise, you cannot have 1 generation in whole life career. So it's really too long times. So now, but in the 1980s, many geneticists, they did lots of studies of age-age genetic correlation from quantitative genetics disciplines. So what we found, progeny test time only need 20%, 25% of rotation age. So that means in radiata pine, we only need age 6. We can select trees reliably at age 6. For Norway spruce, age 12, 15s. So that's been implemented in last 20 years for selection breeding programs. But this for 6, 15 years, that's still too long. Do you think it is too long for such a waiting? Of course, it's too long. So we need to find some other ways to maybe we can shorten in breeding cycles. So we have a dream. Tree breeder, we have a dream for conifer breeding. So I know we Swedish people have a dream. People USA have a dream. Chinese people have a dream. As tree breeders, our dream is we want to reduce progeny testing, maybe much shorter time, hopefully 1 or 2 years. So then plus matings, a few years. So if we can bring 1 generation 5 years, maybe 8 years, then we will be very successful. So we can do 2-3 generations within current 1 or 2 decades for 1 generation. So this is we will pursue this in last 20 years now. So I probably come to this here, people. Yeah. So in the 90s and the early 20s, with the ease of the DNA and screening and mark development, so we were excited. We tried to find significant DNA markers from candidate genes or from random markers. We want to find some markers, like some from genes, maybe some from intergenic areas. We wanted to use marker-edited selections, but we failed. Such methods never been used in tree breeding in the last 20 years. The reason is those markers only capture very small genetic variation compared to the variation we observed from phenotype, from progeny testings. So this one we didn't do well in the last 20 years using marker-edited selections. But since 2000, particularly after 2010s, then because the development of whole genome sequence, first in the animal, they developed this concept is called genomic selections for very early selections. The idea for genomic selection is we selecting on 10,000, maybe 100,000 DNA markers across the entire genome. So we want all those markers cover all those genes responsible for phenotype variation. So like those are genes and the DNA markers. So we have markers across the whole genome, some within genes, some and close within the genes. So with this, if we can have those markers capture 80%, 90%, if 100% sometimes it's more difficult. If we can capture 80%, then actually we can use and apply those genomic selections for very early selections. So we only selection on DNA markers. So we don't need some phenotype for some generations. So this is the ideas for genomic selections. So the process of genomic selections, first step, we need to have a reference population. Some people call training population. Within that reference populations, we need a phenotype, those traits we are interested for breedings. Also, we need a genotype. Mainly, we're using SNPs across genomes. So we got 2 sets of data. Then we put a very big model. We call the mixed linear models, trying to relate all the markers with the phenotype. So that if that model is successful, then in the next generation or any generation related with this training population, we only need a genotype. If we genotype them, we feed in the models, we can predict the breeding values for individuals. That means and the genetic values or the genotype values, so we can make selection. So this is the process. So the process is actually very simple. But with this model building, actually it's lots of, because we have 10,000, 100,000 loci. So this is involved lots of statistics. So why we're interested in genomic selection, the reason is we don't need to wait for 10, 15 years phenotype. So we can genotype at any times, even in the seedling age. So if the model is efficient enough, we can make a selection at earlier seedling age. So then we can realize our dream. We can have many generations and within current 1 conventional generations. So I come to this. Yeah, all thing. So now we can have progeny testing selection at like 1-2 years. So we can realize 1 generation, 5 to 8 years. At this moment, this is still an issue. Some pines, like pines, it's okay. We can have, after selection, 2-3 years, so we get a flower, then we can finish mating 2-3 years. But for Norway spruce, usually take longer. So now we are focusing not only this, also of some of these areas to our and Norway spruce breeding program. So now here, and last couple of years, we did this pilot study trying to find how efficient using genomic selection in Norway spruce. So we selected two forcible progeny at age 28. Within these two progeny, one is nearby Wenden, another is a little bit from here, Hindenburg. So within these two trials, we have 125 families we selected. They come from 55 parents as a forcible cross among them. So we selected about 10 to 20 individuals per families. So we phenotyped them and genotyped about a total 1,400 trees. So we phenotyped 4 important traits for breeding. One is height. Height is always important because it's related to the volumes. We're interested in volume. Another is water density. So also another 2 traits are related with this stiffness of wood. One is MFA, microfibril angle. Another is MOE, modulus of elasticity. So we phenotyped these 4 traits. So those are 3 water traits that we phenotype: water density and MFA. MFA is angle between fiber with this horizontal and vertical lines. If MFA is larger, water is not stiff. So we want to have a small MFA, so have a good wood. Similar things, we use this acoustic method, measure MOE for wood. So now also for genotype, we use the advantage of this Novi spruce genome sequence. In the Novi spruce genome sequence project, we developed this exome capture for design this DNA genotyping. So initially, the project designed 80,000 probes from all 3 kinds of genes: high confidence gene, medium confidence genes, low confidence genes. About half come from this 26,000 high confidence genes. So all those probes come from those gene models. After some initial testing, then we select the 40,000 probes for genotyping. So, and from this 40,000 probes, there's many million SNPs. But after some quality control, so we select about 110 high confident SNPs from those probes. So this is one of results from this genomic selection. We compared relationship among forcible using pedigree based or using our genotyping. For example, in this example, all individuals are all forcible. Theoretically, within forcible, like you with your brother sister, should have 20%, 25% allele same probabilities. But because the sampling process from you to your mother, father, or your sister, brother, they are not exactly. Some brother looks more similar, some are less similar. So we have called this kind of sampling process, we call Mendelian sampling. So that means we cannot capture within pedigree because we don't know, but we use genomic relation, we can capture different individuals. Some are more closely related, some are less closely related. So with this genotype, we can get a much better correlations among individuals. Also, we used this 110,000 SNPs. Then we did this pilot study of genomic selections for 2 sites. So we're using this joint 2 sites for tree height. So we compared if we selection at 28 years using pedigrees, how efficient with the selection. We select the best 1% trees for breedings. So if we using pedigree-based methods, our selection efficiency from 77 for water density to 91% for tree height. So they're around 80% average, 80 to 85%. But now if we don't use any phenotype, we're just using all the genotype. How efficient we select those 4 traits at age 28. So for height, 81% efficient. For water density, 60%. For MFA, 74. So average is about 75%, 76, 75%. So genomic selections, of course, a little bit less efficient than using pedigree. But the issue is here we need to wait for 28 years. Here we can selection at any times if we have a genotype. So we compare the relative efficiency using genotype or using pedigree. So the relative efficiency is about 86, 87. So we can selection anytime. Actually, we can capture 86, 80% genetic gain compared to a selection at age 28. So that's the beauty of the genomic selections. Even though not as high, but we can capture 80%. So we just say if now breeding cycle Norway's blues need 23 years using pedigree-based selections. Now if we can reduce into half and have 2 generations, then within this 23 years, we have 70-80% more gain for height, 71 for water density, for stiffness we have 75. So that's the theory behind the why, and as long as we capture maybe 70-80% this efficiency, genetic variation, then we can do those early selection using genotypes. So I come to this. So this is the genomic selection, early flowering, if we know it's blues. If after, this is conventional, maybe we have progeny testing 15 years, then 6 years mating, 1-2 years. So we have 2 decades for 1 generations. Assume we have a population, mean height 20 years, after 1 generation, 23 years. Then our genetic gain for selections, if we select 10% trees for breeding, genetic gain is 2 meters. So this is our conventional breedings. But this 10% genetic gain usually we can get from 2nd generation, maybe 3rd generations. From 1st generation, we can get more gains, get 20-25%. After that, we normalized. So if now we have 2 generations here, within same period of time, even though using genomic selections, the gain 1 generation is 1.78, less than 2 meters. But if we combine those 2 together, we have more gain, 3.52 meters. So we get like 70-80% more gain per year. So, but if we can like reduce mating times, then probably we can have 2-3 generations, then we have much more gains. So that's the new tool. We're excited for this new eras of breeding for conifers. So at this moment, there's 24 breeding population in Norway spruce across Sweden. So now we're trying to genotype and those population build predictive models for most of these breeding populations. So this is our next step. So to do that, we need to develop a relatively cheaper genotyping and tools. So now we started this international conifer snipper consortiums. So we are develop a snipper array for Norway spruce. So now we will develop a 50K Norway spruce array. We can have a genotype very cheaply for trees. So now we are now have data for whole genome resequence 35 trees also, and so now we are selecting 420 snippers. So we will screening again for 480 trees. We selected from here best 50,000 snippers, build a snipper array. So we can genotype all 24 populations, then become affordable. So then we can implement this genomic selections. So that's the next few years we want to do this. So thank you very much. This is my talk. Yeah.
Thank you very much. Thank you very much, Harry. So any questions for Harry?
Okay. Yeah, welcome. Any questions? Yeah.
I'll start.
Okay.
So Harry, when do you think we can start actually using this for operational tree breeding in Sweden?
I think actually after, because this one, our trial, it's a pilot study, but next step, we are going to get the snipper array by May. So after that, we can screen some of southern populations. So I was already talking to broadcastings about that. So we may be able to start the next 2 years, after 2 years. So after we build this model, they can make selections in the operational levels.
Now you have talked about Norway spruce.
Yeah.
What about Scots pine, the other large conifer in Sweden?
Yes, yeah. So and actually UVA is helping the center. We're trying to now sequence this Scots pines, then also develop a similar SNP array. So actually but it's a little bit behind than Norway spruce. So this is our second object actually goals. We want to also move this to the Scots pines. But at this moment, we don't have lots of genomic information from Scots pine. So we need to sequence Scots pine first.
Yeah, I can comment that. That's actually one of our main strategic goals for the next upcoming years is to launch a big program to also do a full genome sequencing of Scots pine and develop all the same type of tools that we have today for spruce.
Yes.
But it also includes making a much better tools for spruce, I should say, that will make this even more efficient.
Yeah.
Can you comment how much room you think there is for gain? Like how tall can the tree be?
How tall? It's depend the areas. Like I think the trees I saw here, many trees is not tall at all. But it's also depend on species. Like the highest, tallest the conifer species is redwood in California. They can reach 100 meters, but that's take many years. The highest hardwoods is Eucalyptus regnans. They also can reach 100 meters. But most of tree breeding program like eucalyptus, we only get like harvesting 20-25 meter high, because we want usually harvesting value. We don't want to wait. I think for forest management, you have the tools. After the certain volumes, they increase slowly. You have optimal age for harvesting. Yeah. But definitely for Norway spruce, Scots pines, I want those trees taller, because it's not as tall as some species in subtropical pines and the conifers. Yeah. We are also not interested in the size, also the quality. Now that's why last few years we focus on the quality, water density, MOE, but that's very important for our this timber industries.
But can you say something about the maximum gains that have been achieved so far in breeding programs in other tree species?
Like I know radiata pines, our first generation about 30% gain, then a second generation about 11%. I think now this was fourth generations. Actually, it's now more difficult. You say the gain for one individual traits. Now we're using economic, so we're now breeding for several traits, and water density, stiffness, branch size, and also volume. So we have an economic index. So all this, how important each traits to the sawmills, to the pulp mills. Then we have index. We're selecting based on index. Actually, I have some data from radiata pines. Each generation, the values of per hectare increased, but it's not individual traits. I think similar things. Here we want to have bigger volume, but we also have good qualities.
But can you say something about the value per hectare then? How much has that improved from the first generation to the fourth generation?
Yeah, that ones I think. Yeah, I had a figure in another of my presentations. I think at least maybe $2,000 to $3,000 per hectare after the 3 generations in radiata pine increase. But in here I don't know much about this Scots pine and the Norway spruce. So we have some Scots pine breeder and Norway spruce breeder. They could tell me better probably. I think Ulla did some economic analysis a few years ago, had some figures. but I couldn't remember. Yeah.
Last chance for questions.
We're talking about gain, and this method will increase the gain quite rapidly. So generations are getting shorter and shorter, which is great.
Yeah.
But do we have any strategy for maintaining diversity, genetic diversity, which is also very important for the future forests?
Yes, yeah. I have a postdoc, the Henrik. He's doing simulation. So we want to see after the rapid turnover generations how to maintain diversity, like also and the constraint inbreeding levels in the same time to manage genetic gain. This is the issue we need to address. Yeah.
And a comment there is that with these new techniques, you can have a much better control over
The
genetic
inbreeding level.
diversity, right? Because you know exactly how different trees are related to each other, which we haven't known before.
Yeah, yeah.
So
Particularly within families, now we know exactly how they related in the genome structure.
Would climate change will have some kind of effect on the breeding strategy, or
Yeah,
would you like to comment?
yeah. Climate change not only affect genomic selection, also conventional breeding. That's the issue. Actually, Ziqiang now has a project to modeling its climatic effects on the spruce. So we will see when temperature increase 3 degree, 5 degree, how will those breeding population adapt? So actually, we have a huge data sets now, both within Skogsforskning, our collection from long-term provenance trials. Yeah, we will study this. I think definitely climate change will have effects for adaptation of breeding population.
But Sweden is very well prepared for that, right? Since we in spruce, for instance, have these 22 different breeding populations spread out over the whole country. So there is we are kind of prepared to be able to adapt.
Yeah. So spruce breeding strategy like Scotland, they have a bigger mind there for diversity and adaptation. That's why they have so many breeding zones. So,
Okay. If there are no further questions, I think we'll close the session here, and we thank Harry again for a nice talk.
Thank you. Thank you for your coming.